Thank you very much, Anthony, for the kind introduction. And I'm going to present for you today really part of a work in progress. So we're probably about halfway through the story that I'm going to present. So watch this space in about a year's time for its conclusion. And this is on our whole genome sequencing of uveal melanoma. And as Ellen has just elegantly indicated, there are a number of mutations that are known to drive uveal melanoma. And these fall into two main sort of classes, if you like. So down here we have some oncogenic mutations, GNAQ, GNA11, PLCB4, and CYSL-TR2. And these occur really early in the formation of the uveal melanomas. And these actually occur at the stage of the formation of the nevi. But then whether or not the cells then progress to later stages and actually metastasize is due to mutations in genes such as BAP1, SF3B1, or EFAX1 if they're not going to metastasize. And these oncogenic mutations, if you look here in the left-hand side of panel B, these occur in a mutually exclusive manner in the uveal melanomas. And the reason for that is they actually all fit in the same signaling pathway, shown here in panel C. So you have the cell surface receptor downstream of that, GNAQ and GNA11, and they signal through PLCB4. These have produced two really important secondary messages in the cell, and these are diacylglycerol and inositol 3-phosphate. And these then signal to a variety of different downstream molecules. The nature of the signaling downstream for there is not particularly well characterized in uveal melanoma. Regardless of what is published, the things like MAP kinase pathway activation is not really a key driver of uveal melanoma, and you'll hear more in this session on what might be relevant signaling downstream of these oncogenes. These other genes over here, BAP1, SF3B1, and IFAX, they also occur almost in a mutually exclusive fashion, and they really dictate the metastatic potential of the tumours. And once again, as Ellen has just shown you, there's really four classes or subclasses of uveal melanomas which are based on the molecular changes, which are either mutational, point mutational, or copy number changes. And the copy number changes in turn drive gene expression changes which are associated with the good and poor prognosis gene expression profiles that you would have also heard so much about. And the important thing about those four different classes of mutational landscapes of the uveal melanoma is they are highly predictive or associated with the prognostic outcome of those tumors. So if you have no mutations or no copy number changes in anything, basically there is no metastasis. Prognosis is really good. The IFAX mutations are associated with extremely good prognosis, BAP1 with extremely bad prognosis, and then the SF3B1 mutated tumors are somewhere in the middle. And so almost 2 years ago now, we published the first of the papers from the Australian Melanoma Genome Project, and this was looking at the whole genome landscapes of a number of different melanoma subtypes. We looked at acral, mucosal, and cutaneous melanomas, and at that time we did not look at any uveal melanomas, although we had done some whole genome sequencing on a small, about 20-odd cases of uveal melanoma as part of a study that we did when we actually identified the oncogenic PLCB4 mutation. But just looking at the genome-wide mutation rates across the melanoma subtypes shown here, you can see in the middle, the cutaneous melanomas have got a very high mutation burden. This is a log scale on the y-axis, so that acral and mucosal melanomas have about a 25 to 30-fold lower total mutation burden than the cutaneous melanomas. But then the uveal melanomas have actually a 3 to 4-fold lower mutation burden again than the acral and mucosal melanomas. So mutationally, these are very quiet tumours. If you look at the mutation signatures, so this is the context of the single nucleotide changes in those tumours, you can see in the cutaneous melanomas, all this red is mutation signature that's associated with ultraviolet light mutagenesis. You see this in a few of the acral melanoma samples, but you do not see it in the mucosal samples or in the choroidal melanomas that we sequenced in that first group. So we wanted to expand on this and look in a little bit more depth at uveal melanoma, and so we've sequenced now to date another 85 samples using whole genome sequencing. We have about another 30 that are going off for sequencing next week. The majority of these samples are from primary enucleation specimens. We have had some from liver metastases and a couple from short-term cell lines that were propagated from primary tumours. The vast majority of these samples are from the choroid. There's a number of unknown. That's mostly because they come from overseas collaborators, and we haven't been able to source the original pathology reports yet. We've had 3 ciliary body and 5 iris tumours sequenced to date. Now this plot here shows the total mutation burden of single nucleotide changes across the 85 samples. So each column is a different tumour type. So what you'll see towards the right here is the majority of the melanomas have very, very few mutations, in keeping with what we saw previously. But surprisingly, over here on the left, there are a number of samples with extremely high mutation burdens. And if you look at the color key down the bottom here, the black samples are the 5 iris melanomas. All of the 5 iris melanomas have very high mutation burden. There are also 2 other tumors over here that have high mutation burden, and these have mutations in the germline of this gene called MBD4. So what's relevant about MBD4? A year ago, Mark Henri Stern's group published this paper showing that they had a uveal melanoma patient who responded really well to anti-PD-1 therapy, and they had a germline nonsense mutation in MBD4. This is the clinical course for that patient. They had a liver metastasis 9 months after enucleation for the primary tumor. And then they had more disseminated metastases 3 months later. They then went on to anti-PD-1 therapy and had a complete response. And as far as I'm aware, that patient is still currently alive. They then looked, so this is Mark Henri's group, then looked in the TCGA data for additional cases. They found one that had an extremely high mutation burden. They also looked in the other cancer types in TCGA and found one glioblastoma patient that had a high mutation burden. And these patients both had germline MBD4 nonsense mutations. If you look in the middle panel here in B, these are characterized by C to T transitions. And these occur, if you look at the bottom panel here, basically at any trinucleotide contact. So it doesn't matter what the 5' nucleotide is. And this is actually a hallmark of spontaneous deamination of the cytosines. And the reason for that spontaneous deamination is the DNA repair defect, and that DNA repair defect is caused by the lack of function of the glycosylase encoded by MBD4. So we would argue, just looking at this figure here, that there is a proportion of uveal melanoma patients, I would argue all iris melanoma, uveal melanoma patients, and all those with germline MBD4 mutations that should respond well to immunotherapies. And so I feel that these people should be identified and treated accordingly, and maybe even the MBD4 germline status, the screening for that should almost become standard of care, because it does point to clinical benefit in at least that 1 to 2% of patients. But also, if you look down here at the bottom panel for the mutation signatures, in those 2 patients with the MBD4 mutations, you see this dark green here, which is called signature 1, and this signature 1 is the spontaneous deamination signature. And also, importantly, if you look at the iris melanoma patients here, the ones in black, their mutation signatures are dominated by these pink signatures, and the pink signatures are UV mutation signatures. So this, I think, proves once and for all that the front of the eye tumours do get UV, and that's actually driving those tumours. If we look at structural mutations across the samples, again, these tumours are very quiet. There's not a lot of structural changes. We do have the odd sample that seems to be an outlier with high numbers of structural variants, and we're yet to delve into those particular cases to work out what the cause of those more general genomic rearrangements might be. Looking at fusions, a number of cancer types, including some of the forms of cutaneous melanoma, have very specific fusion genes or translocations. We found 2 in-frame fusions in the uveal samples. The genes are listed down the bottom here. None of those are cancer genes in my book, so I don't think they are particularly relevant in terms of driving those tumours. So again, the fusions do occur, but they're probably more incidental than actual drivers. And then we're looking at copy number changes. I'm just listing here the 5 most frequent chromosomes that are altered. So no surprises here with chromosome 3 showing a large number of losses, chromosome 8 showing gains of chromosome 6p and losses of Q. And we do have a number of changes on chromosomes 1 and 16, but the patterns are a little bit irregular. There's gains and there's losses on both of those. So we're actually uncertain at this stage what potential drivers might be residing on chromosomes 1 and 16. So just now looking at the significantly mutated genes in this panel of samples, we see for the first time actually the non-mutually exclusive mutations of GNAQ and GNA11 with 2 of the other, with PLCB4 in this case. But in the 2 samples in which there are both PLCB4 mutations and GNAQ or GNA11 mutations, these are both what we call the weak activating mutations occurring at codon 183. So they're not the stronger mutation occurring at 209. So that might account for the fact that they need 2 drivers here. With one exception, we see that SF3B1 mutations are mutually exclusive with IFAX mutations. And again, with one exception, we see that SF3B1 mutations are mutually exclusive with BAP1 mutations. So future plans, we are doing more samples. We've got another 30 or so yet to go. We need to look much more closely at the structural variants and copy number changes we've identified to look for new potential driver genes. Compare what we get with what we see in the TCGA. But more importantly, if we can find some new drivers, we can then start doing some more functional assays to look at the biology of those. And importantly, not necessarily part of the sequencing project, but part of looking at a larger panel of uveal melanoma cell lines that we are generating or getting from other collaborators. We wanted to do a very large drug screen to look for drugs that might be useful in treating uveal melanoma. And so, as you've heard from Ellen, there isn't any good targeted therapy for uveal melanoma. In theory, all of the oncogenic mutations are targetable, but we would argue that there's no point in targeting, like the cell surface receptor, because most of the samples are activated downstream of it. Probably the best candidate is the enzyme PLCB4, because it's actually the most downstream of all the markers. It should, in theory, be targetable. But then there are a number of other pathways downstream of those effectors that could be targeted. And you will hear more about that in the next couple of talks in this session. And so with that, I just want to thank all the people that actually did this work for me, because I did nothing. And just mostly the members of the AMGP, the Australian Melanoma Genome Project, and a large number of collaborators that supplied the melanoma patient material and information that we used for the sequencing project. So happy to answer questions. Thank you.